Let everyone know that this is being recorded. All right, and so we'll get to our speaker, Jenna Palmisano, who is currently a PhD student at the University of Central Florida. She currently works with rattlesnakes. But prior to this, she received her BS in I think a slightly different area in aquatic and marine biology and environmental studies from Stetson University in DeLand, Florida. And at University of Central Florida, she works in the laboratory of Dr. Anna Savage. And since joining just last year, she's already published 4 papers, if I'm getting this correctly, in Southeastern Naturalist, Microbiology Resource Announcements, Mitochondrial DNA Part B, and Journal of Herpetology, where she is also an associate editor. She has a wealth of experience in herpetology, including 2,000 hours of handling venomous animals, including vipers. I think I got that right. I don't know what I've spent 2,000 hours doing generally, but that's pretty amazing. Her PhD focuses on populations of snakes infected by the disease Raelia, if I got that correct, orientalis. Which was introduced to native snake populations by the invasive Burmese python. And she's also the founder of SLAM, which is the Snake Lungworm Alliance and Monitoring, that networks researchers and managers across the southeastern United States. So today, her talk is titled Using Population Genomics to Evaluate the Influence of an Emerging Pathogen on Pygmy Rattlesnake Populations. And I'll let Jenna take it away.
Okay, awesome. Thank you so much, Robert. Just to fix one of the things you said, I am not an associate editor
Oh,
of Journal of Herpetology. It is Herpetological Review.
I'm
Just
so
didn't want.
sorry.
No, you're fine. I didn't want
Yeah,
to take credit for
yeah. Okay.
something that I'm not. That's all.
Okay, gotcha.
Um, all right. So can everyone see these slides okay? Yes?
Not yet. It is not sharing.
Not yet. Hold on a sec.
Mhm.
All right. Look good?
Now you're good. Great.
Okay, awesome. All right, so thank you so much for everyone that's coming. I know quite a few of you, so hopefully it's not too redundant and you learned something new here. But this will also be recorded, so hopefully we can get some other people to watch it. But the title of my talk today is the impacts of emerging pathogens on pygmy rattlesnake genomic diversity. So this is kind of my goal of my dissertation. So like Robert said, I'm a second year PhD student, so I don't have the results of if there are impacts of these emerging pathogens on pygmies, but I'll set up the context and how I got here and why I'm studying this, why it's important to the research field and the evolution of ectothermic vertebrates. And hopefully I'll answer these questions within the next 4 years about the genomic diversity of pygmy rattlesnakes in the face of 2 major pathogens. So just to outline what we're going to talk about today, I'll talk a little bit about the subspecies of pygmy rattlesnake that is the main focus of my dissertation. So I'll talk a little bit about their life history just to set the stage. There'll probably be a picture of a pygmy on almost every slide because I just got a new camera and I'm pretty proud of the pictures, and they're really darn cute. We'll talk about the threat to these dusky pygmy rattlesnakes, the emerging infectious diseases of concern, which will be snake fungal disease and pentostomiasis. We'll talk about the apparent population declines that are aligned with the arrival of the invasive penostome, and then talk more about my genomic and epidemiological studies with pygmies for my dissertation. So my focal species is Cistoris malarius barbari. This is the dusky pygmy rattlesnake, and I'm focusing primarily on populations that occur in Florida. But there are 3 subspecies of Cistoris malarius. There's the nominate species, so Cistoris malarius malarius, which is called the Carolina pygmy rattlesnake. And then there's also the Western pygmy rattlesnake, which is Cistoris malarius streckeri. And these all occur within like the southeastern quarter of the United States. So the life history of Central Florida pygmies, or Florida pygmies in general, really come from Dr. Terence Farrell and colleagues who've studied one or two populations since about 1990. So there really isn't much more life history information on dusky pygmy rattlesnakes beyond this population. But they are relatively small rattlesnakes. They get to about 36 to 60 centimeters in length and average at 50 to 60 grams, though they do get up to 175 grams. But from the ones that I've captured, the average is more around 45 to 60 grams. One interesting fact about these pit vipers is that they reproduce quite early. They can start mating as soon as 2 years old, which is really interesting, but it's more average. They more likely reproduce when they're 3 or 4 years old. And the average litter size in Florida is about 6, but they can have anywhere between 4 to 12. The Western pygmy rattlesnakes have a larger average litter size of about 10. Their reproductive strategies or history is quite interesting, I think. So they mate primarily in the fall, and then the females will yolk up follicles and store the sperm over the winter, and then they will ovulate in late spring, early July, and fertilize those eggs with the stored sperm, and then they'll give birth in August. So they are live bearing. And they're also ambush predators like most vipers. So you can actually see one in this picture here, tucked in a palm log in ambush. So these guys, these pygmies are mostly eating ectotherm prey. So they're eating lizards and anurans. I actually just found one on Saturday eating a snake, which is a little bit more rare, but they can also eat mammals. They're just mostly eating lizards and frogs. So talking a little bit about the threats to dusky pygmy rattlesnakes, like all wildlife across the world, they are threatened by habitat loss and degradation. And I always use the longleaf pine historic range and its current range as an example of habitat loss, because it includes a lot of pygmy habitat, but you can see how much of it we've lost, with only about 2 to 3% of its original range left. In addition to habitat loss and degradation, pygmies are often killed on the road. So on the bottom picture there, see if you can find the pygmy. I'll circle it in a second. But they're often mistaken for twigs, especially on these dirt roads, and they get hit a lot by cars, unfortunately. So that's where the pygmy is in that picture. So they're pretty easy to miss, and we find lots of roadkill on our surveys, unfortunately. Also, they get predated on by invasive species like feral hogs, pictured here. And unfortunately, they do exhibit human persecution. This is a timber rattlesnake that was killed by a human a couple of weeks ago. So I've found pygmies persecuted. I just haven't taken pictures, though I should. And so this was a colleague of mine who found this timber that was persecuted. So on top of all of these threats, habitat loss, roadkill, human persecution, invasive species predation, pygmies also face emerging infectious pathogens. And the two that we're going to focus on today are snake fungal disease and pentostomiasis. So on the left, you can see an example of necrotic scales from snake fungal disease. This was a snake that I found this past weekend. So this is typical of clinical representation of snake fungal disease, but I haven't molecularly assessed whether or not this was the causative agent for these symptoms, but this is typical to what we see. And then on the right, you can kind of see what the lungworm or the penostome does once the host dies. It typically crawls out of the mouth. So just forewarning that there's going to be some gross pictures throughout this presentation. So just bear with me. So we're going to start with snake fungal disease first. I'll give you some context of the disease as well as when we move into pentostomes, but I'm going to tailor everything I talk about towards pygmies since they are my focal species. So with pygmies, a lot of the time, like I said, you see posterior symptoms. So like in the back half of the body is most often where we see signs of snake fungal disease. And snake fungal disease can really be like super minor, where you just have abnormal scales, like maybe discoloration or displaced, like you can see in this picture on the right here. Or sometimes it can cause severe facial swelling, which is going to hinder the animal's ability to forage. And it can get as bad as these ulcers on the right here and can cause systemic disease in the animals. But again, a lot of the times we're seeing posterior symptoms and things like granulomas or nodules in pygmies. But the disease can get so bad that it can be lethal, but we often see more sublethal impacts in pygmies. So the causative agent of snake fungal disease is Ophidiomyces ophidicola. It feeds on the keratin of snakes, and it causes sublethal and lethal impacts in over 60 species of snakes globally. And on the map here on the right, you can see that it's really, are you monitoring the chat or should I look?
Sorry, I forgot to post the code of conduct. I'm sorry to distract you, but
Oh,
that's
no,
all
you're
that
totally
is.
good. No worries.
Yep.
Okay, no worries. So you can see on this map on the right that the highlighted regions are showing where like the highest prevalence is, and the eastern United States is the area in the world that has the highest prevalence and severity of snake fungal disease. So we know that Ophidiomyces ophidicola has been present in Central Florida pygmy populations since 1992 at least. And it is widespread in Florida. But what we don't know is the strain diversity in Florida pygmy populations and how those different strains relate to severity of disease and whether or not the existing severity scoring systems determine disease outcome. And this will kind of come full circle when we talk about my methods for my dissertation at the end. These are some big questions and big unknowns that I'd like to figure out with my studies. So if you see the picture or the phylogeny on the right, you'll see that there's 3 main clades, and this includes 83 different strains of Ophidiomyces ophidicola. And we're finding that different strains affect different species in different ways. And so I've also published 3 novel genome sequences of Ophidiomyces. So there's just kind of constant work going on genomically to figure out the relation between the strain type and the disease severity. So getting into kind of the sublethal impacts of snake fungal disease in Cystoris malarius, a lot of these studies have been done by Dr. Joe Agugliaro and Dr. Craig Lind, as well as Dr. Terry Farrell. And they've mostly focused on the ecophysiological impacts of snake fungal disease. So they have found that there is increased conspicuous basking, so basking for longer in conspicuous places where predation is likely to increase, and they get a lot hotter, or their temperatures get a lot higher than non-infected snakes. They also exhibit inflammation at the sites of these lesions. They have an increased stress response, so their cortisol levels are higher than snakes that are not infected. And they have altered reproductive cycles, so they're not producing enough the males are not producing enough testosterone at the time when they're supposed to initiate reproductive cycles, and the females aren't producing enough estrogen to go through vitellogenesis, which is yolking up those follicles in the winter. And we're also seeing that they increase their resting metabolic rate, which is kind of relaying the energetic cost of the disease. So this graph on the right is from one of Joe and Craig's study in 2020. And so the open white circles are showing a non-infected snake, and then the black circles filled in are an infected snakes or infected snakes without fever. And then you'll see fever responses at 2 degrees Celsius, increase 4 degrees, increase 5.5. And you'll see that at each level, even from an animal that does not have a fever, their energetic cost or resting metabolic rate is significantly higher than non-infected snakes. So really getting at, okay, maybe there aren't mass mortality events within pygmy rattlesnake populations in Florida, but there are energetic costs to this disease, as well as other physiological costs. So a costly infection could decrease reproduction and extrapolate to population impacts. So what else we're seeing is dysecdysis, so abnormal shedding, emaciation, and anorexia. The difference there is anorexia is the snakes are not feeding any longer or trying to feed, and then an increase in evaporative water loss, which you can see in this graph again. The open circles are uninfected snakes and the closed circles are infected snakes. And there's a significant increase in evaporative water loss in the infected snakes. So if these are the impacts of single infection with snake fungal disease, how do co-infections influence pygmy rattlesnake populations? And this is one of my main questions of my dissertation and kind of leads us into the background for penistomiasis, which is even less known than snake fungal disease. So penistomiasis, the causative agent that causes this disease is Relatilla orientalis. A little redundant there, but you can see it pictured here in the left in a Burmese python's lung, and on the right, a microscopic image of them. So they're quite large. These are both probably about 70 millimeters in length, and they're both females. But just kind of a general background as to what penistomes are, because they are these weird crustaceans we know very little about. So Relatilla orientalis is in this clade known as pentistomes, and they are endoparasitic crustaceans. So they reside in the lungs and upper respiratory tracts of carnivorous vertebrates, and it's mostly reptiles as definitive hosts, but there are some mammals as well. And their closest, penistomes' closest relatives are fish lice, which you can see on the left here. And we call them lungworms because they look like worms. They're vermiform, but they are in fact crustaceans. And you can see 9 different genera on the right here. They all have similar body forms, but the biggest differences are in their head and hook shapes. So all pentistomes are going to go through these 3 life stages: the embryonated egg, which is pretty resilient in different environments. It can withstand desiccation, which is really interesting. And then all pentistomes are going to have a larval stage. The one that we're going to focus on has 3 hosts, but a lot of the pentistomes only have 2 hosts. And we'll talk more about that. And then this is an example of an adult form. So there are native snake penistomes in North America. There's Chirocephalus coarctatus and Porocephalus crotali. And Chirocephalus most often infects water snakes as it uses fish as intermediate hosts, and Porocephalus crotali most often infects pythons, larger colubrids, and vipers as they infect mammals as for intermediate hosts, as you can see in the life cycle on the right. The reason why I put in that 27% of all North American pit vipers have been documented with Porocephalus crotali infections is that leads us to over 70% of North American pit vipers being immunologically naive to these types of parasites, which could exacerbate their response to the disease or response to the infection, which enhances the disease. So we know very little about these pentistomes specifically, so we don't really know their conservation implications. There's not really a clear understanding of their sublethal impacts, and a lot of the prevalence has just been opportunistic. So we know very little about them, but that might be a good thing. It might be that these native pentistomes are just not pathogenic to the point where we're seeing a lot of mortality events because of them. Could just be co-evolved parasites that don't really cause severe disease. And we know even less about the invasive snake pentistome Relatilla orientalis. We do know that it was spilled over with the Burmese pythons who established in South Florida in the early 2000s. And we know this both temporally and spatially, as well as molecularly. So Relatilla orientalis matches the Relatilla orientalis individuals from Southeast Asia. And we started to see them right after the Burmese pythons were established. And the reason why we're so concerned about RO in native snakes is it causes severe infections and has been related to multiple mortality events in different species. And it's also been recognized as a contributing factor to snake population declines in several different species. In addition to population impacts and mortality events, there's obviously a cost of deploying an immune system response. We're seeing a lot of snakes that are infected that are emaciated and anorexic. We see lesions in their lungs when they're related to mortality, or if we get a road-killed snake, as well as scarring in the lungs and inflammation. We see pneumonia, anemia, and sepsis in snakes that are infected. So there's kind of a wide array of impacts on an individual level, and we're already seeing those come out at the population level as well. And in the bottom left, you can see two of the paired hooks. They have these hooks throughout every life stage, in the eggs and the larvae and in the adults, and it helps them attach and move throughout the body. And you can see in the right here, the pentostomes primarily like the anterior portion of the lung, where it's more vascularized than the other parts of the lung. So it's just an example of an extreme infection in a black racer that had to be euthanized. This animal had over 60 pentostomes within its lungs. These are the large ones are all females, and what's moving right now is just their guts. So that's just typical peristalsis. But these specimens were used for, they were flash frozen, and I'm using them for RNA extractions as well as high molecular weight extractions to help us assemble the genome and annotate it. Just more examples of severe infections from Southwest Florida, where the prevalence is extreme. So these are from a banded water snake and a black eraser. So where was RO found in North America? I promise we'll get back to pygmies, but it's important to understand where RO is because it might impact the other subspecies of pygmies soon. So we first found it and still only know of it in wild populations in Florida, but we have found it in one captive snake in Michigan. And we'll talk a little bit more about how the pet trade is going to be able to allow that to transmit further. So in 2019, we knew that RO, I gotta move my Zoom stuff so I can see the map. Before, in 2017, we kind of thought maybe RO was restricted to where the Burmese pythons were, but then we soon found out in 2019 that that was not true, and it had moved quite far, over 150 kilometers north, and we found it in a banded water snake and in pygmy rattlesnakes. And then in 2020, it was for sure outside of the Burmese python invasion front, which is in yellow, which is shaded in yellow here, and the population core, which is in red. So all of these yellow squares are infected snakes. So it's well outside of the python invasion front. So during my undergraduate studies, I wanted to understand how this parasite was moving through the landscape and getting into more native snakes. And so I conducted intermediate host infections experimentally and found that RO has a 3-host life cycle, starting with a coprophagous invertebrate like a cockroach that feeds on the feces of a snake. So the eggs are passed with the feces of a definitive host. And so the cockroaches develop the first stages of infectious larvae. We don't really know how many molts they go through at this point until they're infectious, but we do know that they can stay infectious for over a year. And so the invertebrates then infect the anurans or the lizards, and then those infect the snakes. And there's a question mark next to mammals because relatively little is known about their role in the life cycle, though there's one Kenyan shrew that has been found in the wild with larvae. So just walking you through what each stage of RO looked like. So you can see the hooks in these eggs here. We were able to infect 3 different species of cockroaches, all of which exist in Florida. And this is what the larvae look like, kind of look like a tardigrade, but they're quite large. They're 0.2 to 0.3 millimeters, and they reside in the fat bodies of the cockroaches. And then the cockroaches infect anurans and lizards. And this is an example of one of the lizards that had a heavy infection. You can see all of those little larvae right through the body wall. And we didn't find any health impacts of the larvae, but that is not to say there is none. We just looked at survival and growth rate, and there was not an impact on infected animals within those two variables. And then the snake, like a pygmy rattlesnake, eats a frog or a lizard and then becomes infected with the adults. So the larvae, once they hit the gut, they actually bore through the viscera and get to the lung. So there's likely some cost of that movement to the host as well that we don't know of at this time. So in the wild, Dr. Terry Farrell and I have been looking for infections in native and invasive species. So we found larval infections in all 7 of these species, which include 2 invasive species, which are highlighted in red here. And the reason why they're highlighted is because this is another route for RO to continue its rapid geographic expansion into the southeastern United States. A lot of times the brown anoles and the Cuban tree frogs are moved with humans pretty easily. So another route where RO can keep moving. Snakes in Florida, there's 18 total species that have been found with infection. 14 of those are colubrids. The purple one we just submitted for publication, that's the mud snake. And the bolded ones are also unpublished. Our only elapid has been found. Multiple individuals have been found with infection. And then dusky pygmy rattlesnakes, eastern diamondbacks, and cottonmouths have been found with infections as well. So getting back to my point about immunological naive hosts and what that means for snakes that already experiencing decline, like the southern hognose snake and pygmy rattlesnakes. Well, with pygmies, we think RO is the cause of decline. But the risk factors really are diet. And the issue with that is that most snakes, at least ontogenetically, do feed on ectotherms at some point. So it's likely that RO has the ability to infect any species of snakes. And again, our concern is really the species that are already exhibiting decline outside of RO and that don't have an evolutionary history with snake lungworm species, even the native ones. So for detection, unfortunately, with intermediate hosts, we only know how to detect infection with dissections at this point. So it's a little unreliable. And then with definitive hosts, we use dissections of road-killed snakes, and we also utilize post-mortem movements of RO, like you can see in this picture here. So a lot of the times when snakes die on the road, you can visualize RO moving from the mouth. So if you take a picture, we can typically identify which species of pentastome it is. And then we can also detect infection in live snake through fecal samples. So using microscopy, fecal flotation, and a molecular ID. And the leaders of detection efforts are this monitoring group that I created in the spring of 2022. And that's known as SLAM, or Snake Lungworm Alliance and Monitoring. So since we established in May of 2022, we found 9 new county records, so all the ones in this map that are red here, of infection. And we also found the mud snake infections as well in Southwest Florida. So we have about 50 collaborators across 5 southeastern states, and they're either collecting road-killed specimens for me to dissect, or they are collecting feces from the live snakes that they are permitted to study. Through this collaboration, we are finding more infections in the pet trade, unfortunately. So there's that one in Michigan that was just published by Dr. Terry Farrell, and that was an abandoned water snake. And then I found 3 captive infections, 1 scarlet king snake and 2 garter snakes. And these were from the same breeder slash seller in Miami. And all 3 of these snakes died within 2 months of them being sold. So they were likely wild caught snakes, just based on a couple of them were missing the end of their tails, which is not something that usually happens in captivity. So this is just showing that RO has more transmission routes than just just in wildlife, it's in the pet trade as well. So as we play catch up with this invasion, we really need molecular toolkits to figure out how this parasite or this pathogen is evolving in novel hosts and within its range. So I've assembled and published the mitochondrial genome of RO to help create more diagnostic tools and maybe figure out if there is population structure of these parasites within Florida. And I've also, with my advisor, Dr. Anna Savage, created RO-specific primers. So we're hoping to maybe utilize restriction enzymes with this novel PCR assay so that other labs can test for the parasite without having to pay for sequencing. And so right now, this image of my gel is just showing that our primers are specific to RO, with the positive control being on the left. And on the right, there's its congener, Relaitiella indica or frenatus, they're synonymous, and then a native penistome. So they're either getting really high off-target bands or no bands at all. So we're hoping to utilize restriction enzymes to validate this method further and then publish it for other people to use. Okay, so now kind of getting into the bulk of my dissertation studies on Cystoris malarius and the declines that we're seeing with the first documentations of RO prevalence. So in Florida, this has been known, Cystoris malarius has been known as the most common venomous snake, but in South and Central Florida, they are rapidly disappearing. So one example of this are records from the Everglades National Park road surveys. And so Dalrymple did a lot of studies in the 80s where he cruised up and down the park and he found thousands of snakes, and about 11% of them were pygmy rattlesnakes. And in subsequent decades and in subsequent years, that number has dramatically declined. So in 2009, and keep in mind, Burmese pythons were recognized as established and reproducing in 2000. So that was likely when the initial spillover event occurred. But in 2009, the percentage of snakes caught that were pygmies was below 5%. And then it dropped to below 2%. And now it's less than 1%. So in the Everglades National Park, which is one of my study sites, there has definitely been major declines and evidence of major declines in pygmy rattlesnakes. This is just some initial energetic modeling efforts. It's using the models that we've created for snake fungal disease to assess the energetic costs of disease. But the issue here is that we haven't done respirometry trials with penistome infected snakes. So what this model, this simulation is taking into account for this orange line here is just the energy that penistomes use. So that's how we're calculating the cost in this graph, but it's probably a conservative measure at this point because there's probably a lot more energy taken or given by the host to respond to this infection. So this is just some preliminary results, but we're hoping to upgrade this model. Also, I'm working now to translate it from macro code to R script because I don't want to work in Excel anymore. So from all of this, we're seeing that RO is a major conservation concern for pygmies and other species. It's using widespread, intermediate, and definitive hosts. Its geographic spread is rapid, and there's really no known limitations. And we're seeing that pygmy declines are aligned with the first detections of RO. And just last year, I found 6 dead individuals between 2 of my mark recapture sites, and all of them had infections. And we just don't understand the impacts of co-infection with snake fungal disease. So for my statewide studies, I'm studying 12 populations, probably will turn that into 13. But I am surveying for Ophidiomyces ophidicola, so the causative agent of snake fungal disease, and Relaitiella orientalis, the causative agent of pentostomiasis. In addition to my epidemiological studies, I'm also taking genetic samples. So I take scale clips to assess the neutral diversity within and between populations. And I'll be using a DD-RAD-seq approach to do that. And then to look at adaptive immune alleles, I'll be using an amplicon sequencing approach to understand the diversity of MHC class 2 alleles within in between populations. And just to go over kind of my questions from the statewide assessment is my questions and predictions. So for predictions, populations recovering from RO invasion are likely to have higher frequencies of the advantageous MHC alleles and lower genetic diversity. And that is, of course, if we see a trend in MHC allelic diversity or certain alleles correlated with disease severity or prevalence. And so a subset of those locations, I will be using comparative museum samples, so 3 that are RO positive and 3 that are RO negative, to assess pathogen-induced bottlenecks. Another prediction I have is that genetic differentiation will be high among S. malarius populations, in that there's likely more than 3 distinct genetic clusters. So the only genetic work that's been done on dusky pygmies show that there are 3 distinct clusters: the Everglades, the Peninsula, and then west of the Apalachicola River in the Panhandle. But I think with more fine-scale data and a higher sample size, that we are going to find that there is more genetic structure between these populations. And some questions I have about ophidiomyces are things that I've already mentioned, is that, I want to understand if there are different strains and if they are related to severity that we're seeing across these populations. So out of 3 out of those 12 populations, I'm studying those pretty heavily. So I go out 3 days each month, and I'll do that for the next 4 years. And each of these populations have varied initial detection dates of RO, and they have varied prevalence at each site right now. So I think it'll give us a really good idea of the impacts on survival, growth rate, in different demographic groups. So with that, my questions are, our predictions are that, larger snakes are likely to have a higher prevalence of RO based on consuming larger prey items and more prey items. So they just have a higher chance of getting infected. Infected snakes will have lower survival and decreased growth rate. And one of my questions is, do current SFD scoring indices relate to the outcome of disease? So right now there's 3 published scoring ways or scoring mechanisms, and I want to understand if any of them actually relate to the outcome of disease or the survival. So far since June, since I heard about the SSE grant, I've done a lot of field work and a lot of lab work. I've caught over 170 individual pygmy rattlesnakes across 11 populations. So I still have 1 population to go to. 5 of those 11 have RO prevalence, which is more than I would have liked. And 7 have clinical representation of snake fungal disease. And keep in mind, we often see the highest clinical symptoms of SFD in the winter and spring months, and I've only studied them in the summer so far. For the mark recapture sites, there's 3 kind of different levels of prevalence, and I haven't assessed all of my fecal samples just yet, so I don't have actual numbers for you. But each of these populations, from what I have assessed, I would grade them as low, medium, and high RO prevalence. And the one that's high has actually exhibited decline since 2016, when we first detected RO at this population. And we've definitely seen a crash where we used to be able to get tens of snakes in one day, and now we struggle to get 1 or 2 snakes there. And so at that site, I've had 11 total captures and 1 recapture. At the medium RO prevalence site, I have 49 captures and 5 recaptures. And at the low RO prevalence site, I have 51 captures and 12 recaptures. And I'd like to just say that through all of these sites, they all have the same amount of effort. So I think it's really telling that the high RO prevalence site is exhibiting a decline, as we used to be able to get a lot more snakes there. And with my lab mate, we just completed a pilot sequencing run for MHC class 2 across half of my population. So stay tuned for results from that. And just kind of ending on a higher point, there are still really big pygmies out there. I found this giant pygmy, which was 175 grams a couple weekends ago in the Everglades. So hopefully there is a chance for pygmies to bounce back from genetic bottleneck if that is indeed what is happening. Just kind of a takeaway from this whole talk is that if you see dead snakes or moribund snakes, you can contact the Herp disease alert system, which is through Partners in Amphibian and Reptile Disease. So you can get there through this fact sheet QR code, which is on the bottom left. And then you can also contact me, and I have my email on the next slide. If you see any snakes that are dead with worms crawling out of their mouths, please come contact me. And then this is just a QR code for my Twitter where I post a lot of the pygmy research. And then I just like to say thank you to all of my funders, especially the Society for the Study of Evolution. It's a huge honor to be funded by y'all. And with that, I'll take any questions.
All right, great. Thanks so much, Jenna. Really great talk, fantastic visuals. Uh, and yes, people are saying great talk in the chat. Anybody has any questions, you can feel free to raise your hand or turn your mic on or video or type in the chat. And if not, I'll start it off. So is the pathogen really severe to the Burmese python or like other snakes in its native range?
I'm so glad that you asked that. That was one thing I forgot to say. So comparatively, no, these pentostomes RO gets much larger in native snakes compared to Burmese pythons. So the females are reaching much larger sizes, and the infections are a lot more severe. So you see way lower numbers in Burmese pythons compared to native snakes, where native snakes you can see over 100 pentostomes within the lungs, and with Burmese pythons, you typically see like 5 to 12, and they're a lot smaller. And we don't really know much about RO within its native range.
So the Burmese has some defense then, presumably to prevent. Yeah, and it seems like the maybe RO benefits from that defense that the Burmese python has because it allows the Burmese python to then live longer and produce more eggs. Is it advantageous for the RO to kill the snake?
I don't see why it would be unless there was another host that they could get into, like if a hawk or a vulture was able to, it was
Hmm.
susceptible to the disease.
Mhm.
Um, Yeah, I mean, otherwise, there's kind of that optimum when it comes to parasites
Mhm.
between virulence and transmission. So you wouldn't want, yeah, I don't know. I don't think it would be advantageous to kill your host in this aspect, but it could be, especially maybe they're evolving to get into scavengers that are going to eat dead snakes, and maybe that's one of the reasons why they have the propensity to move out of the mouth. But I more so think that that's due to their aerobic respiration. But yeah, that was a long-winded answer of I don't know.
No, thanks for that answer because I was wondering what is the goal of that RO that's coming out of the mouth of the snake? Like where is it trying to go to?
Yeah, my best guess is that it's the oxygen depletion because they do aerobically respire, so they're just trying to get out. But it's weird because we don't see that type of post-mortem movement with the native snake penostomes. Um, So I don't know.
Hmm.
I think there's all different types of questions you could ask, and there's always weird dynamics between a pathogen and its novel host.
All right, I'll let if anybody else has a question, I'll let anybody else ask.
That's all right. They have my email if they need if they have any more
No
questions.
problem. I
Send
have
them.
I have a laundry list here, but I'll probably just ask one more, and I'm trying to choose which one.
I mean, we
Uh,
have 9 minutes. We have 9 minutes. You can ask
yeah,
as many as,
true, true. Good point. Um,
yeah.
yeah, I was gonna ask. It's if I got this right, the Burmese python was introduced in 2000, and the first, that's right.
So
Mhm.
they were recognized as established, so they were recognized as like
Oh, I see.
reproducing
Hmm.
populations in Florida in 2000, but I think the first like initial sightings of Burmese pythons were in the 90s.
Oh, wow, so even earlier. Okay. So because the disease was detected in 2017, right?
So, oddly enough, that
Mhm.
study does not tell us when the first detection was. All they
Hmm.
say within that publication and within the dissertation that I've read many times is that they collected snakes from 2004 to 2017,
Mhm.
but it doesn't say when their first infected snake was found.
Ah.
So, I don't know. Presumably, it was prior to 2017.
Mhm. Okay, got you. Because I was just thinking that was a long time
Right.
for maybe it was under the radar, or maybe it wasn't in the initial Burmese python population and then got introduced by one later, or.
Yeah. So I mean, I kind of have several hypotheses to that,
Hmm.
and I'm hoping with my genomics work with pentostomes I can answer that.
Hmm.
But I think that there is a likelihood of multiple invasion fronts of RO because it moves with cockroaches, it moves with a bunch of different invasives. It's in tokay geckos, tegus. So there's likely more than one invasion pathway. But I do think just based on how many Burmese pythons were able to establish that they were the likely first introduction. But I mean,
Hmm.
it could be a case of like it takes a while for a pathogen to like permeate an ecosystem. So maybe it's not until we see it saturate the ecosystem that there's a
Hmm.
problem. So it kind of could go under the radar, but I'm not sure we understand the dynamics enough to answer that. But
Got you, got you.
yeah.
Uh, yeah, I was just thinking because 17, like I think it could be under the radar, but 17 years is a long time, but that makes sense that the dates weren't completely accurate, so it's a little
Right.
uncertain.
Yeah, so I've asked the researcher a couple of times and I haven't gotten a very like definitive answer as to when they first detected them, but my guess is before 2010.
Got you.
Yeah.
Okay. And then okay, I guess this can be possibly my last one. But it's like a doomsday question. Like, is there anything limiting the spread of this pathogen of RO? Will we expect it to go all across North America, or maybe there's some winter temperature that limits it or something?
Yeah. We don't know of any limitations at this point. We know that
Uh-oh.
if we know that if the eggs, the larvae, or the adults are frozen,
Hmm.
then they get killed.
Hmm.
But they do survive complete desiccation. So yeah, we don't know all of the conditions um with like which the eggs can survive. So I can't answer that definitively, unfortunately. But at this point, we don't see any restriction because the intermediate hosts are widespread and abundant, and it's kind of a super generalist where it
Yeah.
can utilize invertebrates, it can utilize all different types of frogs, toads, lizards, and we know it can utilize mammals. We just don't know how much or how often, or if that's a paratenic host, so one that the larvae doesn't develop in but
Mhm.
can transmit through. And then with the pet trade and stuff, and people keeping animals outside, people releasing animals, yeah, I think there's a high potential for this parasite to spread across the country, and potentially across the continent. Um, I think, yeah, even like with freezing being maybe a limiting factor, if it's within a host, it's that host
Oh
is
yeah.
likely to burrow and get out of the frost line and probably will be fine within the host, but
Mhm.
eggs probably wouldn't make it through a freeze.
Okay, and actually, okay, this is the last one. I promise. This is more of like an applied question. Like, so I'm in North Carolina.
Yes.
Is there value to contributing to the network outside of Florida where the pathogen is?
Yeah, absolutely. Yeah, we have several collaborators in North Carolina, South Carolina, Georgia, Alabama. So we have quite a few people looking at dead snakes for this invasive pentostome. So yes, I do think that it's worth it, just because what we're seeing right now is such like a sporadic expansion of its geographic range, just based on like it being able to use so many different hosts. So and a lot of the hosts are synanthropic, so they like are around humans a lot or might be moved by humans, like especially like big RVs or other things. Like curly tails get moved to Georgia all the time, with like lumber movements, and then through horticulture as well, there's a bunch of Cuban tree frogs and cockroaches moving up and down the coast. So I would definitely say it is worth it to look throughout the Southeast, because I think even negative data down the line will be
Hmm.
important, because I do think that at one point RO is going to reach these states, and with all the threats that snakes and other herpetofauna already face, this could be a huge contributing factor to continued or um, maybe even advanced population declines.
All right, well, I will keep my eyes open while I'm on the road then. In that case, I'll pull over.
Cool.
Uh, so thanks so much, Jenna. Thank you everyone for being here and staying for the question session. And we will see you next week for the next Greg seminar. And thanks so much, Jenna.
You're welcome. Thanks for joining and having me.
All right.